so that. Thank you, Iman. Yes, dear, I'm fine. Alhamdulillah. If you remember, I told you that last time I'm going to discuss the because as you remember, I told you that I usually do an analysis. After the midterm is over, like which question was basically most of the students. If I just try to find out these little statistics. Which question had a major problem with most of the students, and I found two questions. Okay. And I really wanted to discuss those questions here before any other midterm, before the final exam, or before, for even if we don't give these questions in the final exams, still I wanted to clarify what exactly happened with the question. This was a question which most of the student, they did a mistake in this. Tell me why, what happened? Like, this was like the most easiest question. I mean, there was nothing in this question. It's just that there was a slide in the presentation where where you had to memorize if it's an naproxen. Okay, the S enantiomer is the painkiller, while as the R enantiomer is the liver toxin. Can anyone tell me what exactly happened with this question? Why most of you? I'm not saying you guys. Maybe you're not one among those. It's like the analysis is basically based on uh, everybody, Mr. Muhammad's students as well as my students. So what was the problem with this question, guys, exactly? Anybody wants to share your reviews? I don't remember the statistics, but I think I just had 54 students out of so many who actually got this question correct. Can you believe? I was like, why? So everybody is silent. Nobody's answering. Yes, Yomna, Muhammad, Iman, Faiza, who will answer this question? What happened with this question? Rand, Tashil, Tanya, did you kind of ignored this question? You thought of maybe this is not important. I think because our concept R is not. Maybe we didn't focus. Okay, no problem. Okay, thank you for accepting that. Maybe we didn't focus. Yeah, it happens. Sometimes you think that, oh, this question is so easy, like Alhamdulillah, good. Yeah, as I said that, there were very, very few people, who got this question right. I gave you the statistic, like I think 53, that's all. That was a very less number. I remember I viewed the recording, so I saw you saying it's important. Really? Did I? I mean, yes, whenever I feel like, you see, when the questions are given, maybe when I was teaching in the class, and maybe I said that, because when the questions are given, as you remember, you should remember this forever. I tell this in PHSF as well, okay? You should touch each and every concept, because whenever, for example, we make a paper, we try to touch each and every concept, right? So that is why don't think that this is important, this is unimportant. Yeah, it was a small topic, absolutely. You just ignored it. I can totally understand it. Okay, good, Alhamdulillah. But anyways, so this was like there was no concept involved here. It was just about memorization. And to my surprise, the another question which most of the people got it wrong was this one. Guys, what happened to this question? Which of the following statements is correct about the glucose? Don't you remember we discussing this slide like C4 epimer of galactose? Look at this. Didn't we discuss this slide in the class? Glucose is the C4 epimer of galactose and glucose is the C2 epimer of the mannose, right? And don't you remember when we said that? See, it was very important to know, you have to clear your basics. When you start with the carbohydrate chapter, the first thing which you all must know that when we say aldoses and when we say ketoses, so it should be very, very clear, if not other structures, but the basic structures like glucose and fructose, right? You should know that glucose is an aldose, okay, which has an aldehyde group, and fructose is a ketose with the C double bond O at the position number 2, right? So if you see here, so the certain things which are very, very, the basics should be very clear. If you remember, there was this slide which I had put, in my presentation, right? So if you see here, if you see the aldehyde group, okay, whether it's a pentose, whether it's an hexose, whether it's a triose sugar, the moment you see aldehyde group, that means it's an aldose. The moment you see the carbonyl group at the position number 2, that means it's a ketose, right? This is something which is very, very basic. Okay. So for example, glyceraldehyde, if I ask you a question about glyceraldehyde, what is it? It's an aldose or a ketose? The glyceraldehyde, the triose sugar, it's an aldose. We thought that we will get a diagram and we'll identify it. Okay, but that's okay, right? So that's why, we should always start with the scratch. The scratches you should very, for the final exams also, be very clear about which one is aldehyde, which one is ketose. Okay, no need to remember all the structures, but the basic structure, like at least the glyceraldehyde, the hydroxyacetone, glucose, fructose, pentoses, these are like the basic structures which at least you should know how to identify. Nobody's going to tell you to write these structures, but at least you should know how to identify if it is an aldehyde, as an aldose, right? So maybe that went wrong here. It's a ketose sugar. Maybe nobody knew what is glucose. Whether it's a ketose or a glucose. And then, yes, what about this? This was so very clear. If you remember when we discussed the carbohydrates, I told you that D form of, monosaccharides, or, your carbohydrates are the only ones which are present in our system, right? Isn't it? So that was basically, if you remember, the D form is, the D form basically wherein you have the OH in the penultimate carbon, right? Or the carbon with the highest number of stereo center, right? So yes, so remember, it is basically the D form, okay? And especially for the amino acids. For the amino acids, what is it? Is it D form or the L form in the biological system? Which one is more common for the amino acids? Really? Abdulaziz, I want you to think and get back to me again. Study your amino acids and tell me, in the biological system in our body, what kind of amino acids do we have? Okay. I want you to get this straight. This is very, very important to know, right? These things are very, very important, right? So I want you to study the proteins chapter, and then I'll get back to you on this. Okay. So these were the two questions which were basically, which were kind of, but that's okay. These were the only two questions, right? It's just everybody, mashallah, the overall, alhamdulillah, the result was good, alhamdulillah. So let's try to upload today's chapter. Give me a minute. Okay. So we started with the structure of DNA in the previous class. We started with the primary structure of DNA, right? So then we said that in the primary structure, we said that it's basically the sequence of nucleotides, right? Which is very, very important when we talk about primary structure of DNA, right? And we said that we always read the DNA strand from 5' to 3' end, right? The way we read alphabets in English, we start from left to right, right? So similarly for the DNA, the standard is like 5' to 3' end, okay? Then we had to start this one. Okay. Then the primary structure can further be divided into the if we talk about the DNA structure. Okay, so we can say that the backbone of the DNA, this one, okay, basically it has an alternate phosphate and the sugar, phosphate, sugar, phosphate, sugar. So this forms the backbone of the DNA. Okay. So what if the question says what is present in the backbone of the DNA? So you will say that's just the phosphate and the sugar, no bases. Bases are not present in the backbone. They are present towards the inner side, as you can see here. They're towards the inner side. Okay, so bases inwards. Okay, and sugar phosphate, they are present outwards. Okay, because outside we have the hydrophilic environment. Okay, if you remember, in the plasma membrane we spoke that the hydrophilic heads are from the outside and the hydrophobic tails are from the inside, right? Because outside in this, we have what? hydrophilic environment. Same thing here. Okay, here, because of the phosphate, it is polar molecule, right? So the hydrophilic part is from the outer side, okay, where we have the water, because in the cell you have water everywhere, okay, even in the nucleus, okay. And bases are hydrophobic, so they are present towards the inner side, the way tails are present towards the inner side. Just remember this again, okay. So then what stabilizes the two strands of the DNA? If I have another strand of DNA over here, what stabilizes the two strands of DNA? That is basically one, the base pairs. For example, guanine and cytosine is here, right? So we have three bonds between them, three hydrogen bonds. So again, three hydrogen bonds over here and cytosine over here, right? So one, these plenty of hydrogen bonds which are present between the bases, they stabilize the structure of the DNA. And other than that, the hydrophobic interactions present between the bases. Because as you can see here, bases are stacked together, isn't it? The way you have, for example, if I give you 10, 20 coins, you'll put one coin here, another coin on top of it, another coin on top of it, another coin on top of it, right? So the way coins are stacked together, similarly, these bases are stacked together, right? So the hydrophobic interactions also stabilize the, also when I say stabilize, what does that mean? The question says that, for example, the question says that, what holds the two DNA strands together? Okay, we know that the DNA strands are something like this. This is a helical structure, right? So they are never separated from each other. They are together, aren't they? They are together, right? What keeps them to stay together? Why are they together? Why they don't separate out like this? Right? Why they don't separate out like this, right? They're always together. So the question says, what stabilizes the DNA structure? It used to be only hydrogen bonding once upon a time, but recently they have also discovered that it's not only the base pairing between the adenine and guanine and cytosine, and adenine and thymine. It is also the hydrophobic interactions present between the nitrogenous bases. Okay. So that is also very, very important, which holds the DNA helices, two strands of the DNA together. Okay. So they are glued together by two things. One, the hydrophobic interactions, and number two, by the base pairing between the bases. Okay. So because we know that these bases, since they're hydrophobic, they try to stay away from the water. They try to hide themselves from the water. So that is why they face each other, the way we spoke about in the plasma membrane, right? The tails, they face each other, right? Similarly here, since they want to stay away from the water, so these bases are stacked together. So hydrophobic interactions as well as the base pairing stabilizes the DNA. But it's mostly hydrophobic interactions than the base pairing. Yeah, yes, Mohammed. Yeah, that's my next thing. First of all, I wanted to explain the, what holds the two strands together. We'll go one by one. Don't worry. Okay, I will explain that. Okay, that's in the upcoming slide. So, are we okay about what stabilizes the two strands of the DNA? Any questions about it? Okay, then I'm not going to discuss this because we already discussed the difference between the DNA and the RNA previously, right? Here it's trying just to show you that what is the difference between the backbone of the DNA and the backbone of the RNA. Of course, in the DNA, you will have the deoxyribose sugar, right? And in the RNA, you will have the ribose sugar. This will be the first difference between the two, right? And next, we already know that in the RNA, we have the base uracil, and in the DNA, we have the thymidine, right? Now, this I'm going to explain you in the next slide, okay? The directionality of a particular DNA. Now, if you remember, I told you last time, the two strands of DNA are basically, for example, your 5 dash, 3 dash, right? And another one is anti-parallel, right? Another one will be 5 dash, 3 dash, isn't it? If you remember, I gave you an example of a traffic coming, you know, two-sided traffic. Okay, one side, the Mariam cannot hear me. What about others? Can you all hear me, rest of the students? Mariam, maybe you have a problem with your headphones or your computer. Please check your system, Mariam. Okay, thank you. Thank you for confirming, everyone. Okay, so if you remember, I told it's like something like a, two-way traffic. You have a divider in between. The cars are either going towards the Al Faisal University, and the other cars are coming out of the Al Faisal University, right? So they are anti-parallel. The strands are anti-parallel. Yes, Mariam, you can. Absolutely. You can. Okay, no problem. No problem. You can join with the other device. No problem. Okay, now Mohammed asked me a question. Now the question is that what is this 5 dash and 3 dash? Dash, you already know that. I explained you when we use the word dash, it should automatically come in your mind that she's referring something about sugar, right? Remember the bases, we never use the word, dash for the bases, right? We simply write as 1, 2, 3, 4, right? We discussed that previously. Now, the moment when we say 5 dash and 3 dash, what exactly does that mean? If you look at this is one strand of DNA, and and there's no other strand, there's only one strand here. But anyways, it's the same thing on the other one as well. So this is the sugar, right? This is the sugar. Okay, this is the glycosidic bond between the anomeric carbon and here between the base. We discussed all that previously, right? But look at this. If you see here, when I say 5 dash, that means the carbon at one end of the DNA strand, right? The carbon of the sugar, okay, please pay attention, the carbon of the sugar is attached to the phosphate like this. So at the 5 dash end, you know that it is the phosphate which is attached to the carbon number 5 of the sugar. That is why we say the 5 dash end, right? Then if you see here, we have base, sugar, phosphate, base, sugar, phosphate, another nucleotide, base, sugar, phosphate. So in the backbone, you have alternate phosphate, sugar, phosphate, sugar, phosphate, sugar. And now if I go to the other end of this strand, what do I see? Can anyone tell me what can we see at the other end? What do we have? We have at position number 3 of the sugar, we have the hydroxyl group attached to it. Is it clear? So in the pentose sugar, we have the hydroxyl group attached to the 3 dash carbon. So that is what is whenever we say 5 dash and 3 dash. Okay, so 5 dash is basically this one where you have the phosphate attached, and 3 dash is the third carbon of the sugar where you have the hydroxyl attached. Mohammed, is it clear till now? So what is the meaning of 5 dash and the 3 dash? Is it clear? Yes. Okay, alhamdulillah, right? And you will see once we'll discuss the replication of the DNA. So this 5 dash 3 dash is very important because you will see that once we will discuss the DNA replication, you will see that if you have to make this DNA chain long, the nucleotides will always be added at this 3 dash end. Remember this forever. If you have to make this DNA chain long, if you have to extend this DNA chain, the new nucleotides are always added at this 3 dash end, right? We'll discuss that once again when we will discuss the replication. Okay, clear? 3 dash always has OH and 5 has always phosphate. Absolutely. Yes, Abdulaziz, you're right. Dana, I will repeat once again. Don't worry, Dana, I'll repeat. I simply said that what is the meaning of 5 dash and 3 dash? When we say 5 dash and 3 dash, you just saw here, I told you that one strand of the DNA is from 5 to 3 prime, and another strand is from the other way, from 5 to 3 prime, right? They're anti-parallel. Okay. Then Mohammed asked me a question, what is the meaning of this 5 dash and 3 dash, right? So 5 dash, if you see here, let me erase this all. If you see here at the 5 dash position of my sugar, right? I have a phosphate group attached to it. So this is my 5' end. Because if you remember, I told you that sugars are always named by we add a small dash over here. So we call it a prime, right? So it means that at the 5' of the sugar, the phosphate is attached. And on the other hand, when I say it is, if this is the DNA, so we say 5' and we say 3', right? So at the other end, at the position number 3 of my pentose sugar, I have the OH attached here. That is what it means. Okay, Dana? So DNA has a directionality, right? And then how, if the question says that, if the question says that how are your sugar and phosphates attached to each other? Okay, if you remember, we spoke about an ester bond, right? The phosphate is attached to the sugar by the ester bond, right? So if you can see here, look at this. We have an ester bond over here. We have an ester bond over here. Isn't it? Is it clear? So we have a phosphodiester bond, right? So phosphate is attached to the sugar, right? Alternately, as you can see here, we have a phosphate and sugar, then phosphate and sugar. So it's attached by the phosphodiester bond. Okay, so what is a phosphodiester bond? Phosphodiester bond is a bond between the phosphate group, as you can see here, and the sugar in a polynucleotide. This is a polynucleotide with many nucleotides over here. Okay, so the question says that name the bond which links the two sugars of the nucleic acid. Okay, so you will say it is the phosphodiester bond. It connects the 5' carbon, as you can see here. Let me erase this. It connects the 5' carbon, okay, of one nucleotide to the 3' carbon of another nucleotide, because this is my one nucleotide, correct? And then this is my another nucleotide, right? So they are connected by the phosphodiester bond. Again, a very important thing. Okay, so if if the question says that name the bond which links the two sugars, or name the bond, okay, which connects the one sugar with the other phosphate, so you will say it is the phosphodiester bond. Like these are the basic things, which are basically, which can be asked, okay, about which you might ignore. You might think that, oh, this is not, knowing the bond is not important, or knowing what is present in the in the sugar phosphate backbone, okay, or to which side the bases are present. Are they present inwards or outwards? Okay, or what is the directionality of the DNA, right? All these things are important when it comes to the structure of the DNA. Okay, as you can see here, this is the 5' end. Now you all can understand this when I say 5' 3' you all can relate it now. Look at this. This 5' here, it has a phosphate group attached here. At 3' it has the OH group attached over here. Is it clear to everyone? And look at the hydrogen bonds, the 3 hydrogen bonds between the guanine cytosine, and 2 hydrogen bonds between the adenine and thymine, right? So we were talking about the primary structure. So thus the primary structure of DNA is the order of what? Nucleotides present in the chain, or the order of bases present in the chain, which are from 5' to 3' direction in the one chain, and from 3 to 5 in the other chain, right? Now the next thing is the Chargaff's rule. Okay, there was a scientist, his name was Erwin Chargaff, okay, in 1905, and he received a Nobel Prize also for this. Okay, he said that the number of adenine, okay, in one strand, or the number of moles of adenine in one strand, is equal to the number of thymine in the other strand. Do you all understand what I'm trying to say? If you look at this table once again, look at this. Adenine is 30.9. What about thymine? It's again 29.4, which is pretty much close to 30 if you take a round figure. So number of adenine is equal to the number of thymine, Okay, this was basically, yeah, this was basically said by the Irwin Chargaff. We call it a Chargaff's rule, right? And if you look at the guanine and the cytosine, okay, look at this. Here we have 19.9, and this is again almost same. So guanine is equal to the cytosine. The number of guanine moles in one strand of the DNA will be equal to the number of cytosine in the other strand. So this was called as a Chargaff's rule. Is it clear? And based on this Chargaff's rule, okay, the two important scientists, right, the Watson and the Crick, who gave us the model of the DNA, right? The DNA, the model of the DNA was given by Watson and Crick, right? They received a Nobel Prize for this, right? So they proposed the double helical model of the DNA, which is the secondary structure of the DNA, based on the information what was given to them by the Chargaff. Okay, so in the secondary structure of DNA, what do we say? We say that the ordered arrangement of the nucleic acid strands, okay, and how they're coiled around each other, okay, gives us the secondary structure of DNA. Okay, so this, how the coils are arranged to each other, how adenine is, how adenine binds to the thymine or guanine to cytosine, right? So this was basically very, very important discovery. Yes, you should be able to know. You don't need to know the table. You don't need to memorize the table. Just remember the compositions, right? The adenine is equal to thymine and the guanine is equal to cytosine. That is important to memorize, right? So, so as per, just that they're equal. Yes, Fatima. The concentration is equal in both the strands. Absolutely, Fatima. Yes. Right. So according to the Watson and the Crick model of the DNA, they said that the DNA is double-stranded, double helical structure. As you know that, it's not a straight like this. It has a helical structure, right? It has a helical structure like this. Okay, it consists of two chains of nucleotides, as we already know about it, right? And the two polypeptide chains are always anti-parallel. We discussed that in a while, right? 5 to 3 and 3 to 5, running in the opposite directions, connected by the hydrogen bonds. We discussed also that, like between adenine and thymine, we have two hydrogen bonds. Between guanine and cytosine, we have three hydrogen bonds, right? And DNA is made up of small subunits called as the nucleotides. So biggest mystery was solved by when Watson and Crick gave their model of DNA. Right? And then they also said that the two strands are not spaced equally. Right? For example, if you see here the strand, the strands are equally spaced, but they are not actually equally spaced. As per the Watson and Crick model of DNA, as per its secondary structure, they say that the way they are coiled to each other, it results in the formation of a big groove or a big space. As you can see, the purple one over here, this big space over here. Okay, this is called as a major groove. Okay, and then here the helical structure is not equally spaced, right? And here it forms a very smaller space, which is called as a minor groove, right? So this was also given by Watson and Crick, that they are not equally spaced. Okay, so they have a major groove as well as they have a minor groove. They also said many other things, but whatever is in your book, we'll just concentrate only that, will not go beyond that. Okay, and this we already discussed. They are an anti-parallel, right? We discussed all this. This is again, we discussed all this, the hydrophobic interactions, we discussed all that. Again, this we discussed all this, like between adenine and thymine, you have two hydrogen bonds, and you have three hydrogen bonds between guanine and cytosine. And there's a complementary base pairing, remember. For every adenine on the other side, you will always have thymine. For every guanine on the other side, you will always have cytosine. You will never have adenine and cytosine. Remember that, right? The base pairing rule. The DNA follows the base pairing rule, right? That was all about the structure of the DNA, the primary and the secondary. But we'll not stop over here, because we know that that is not how DNA is present when it comes to the what? Cell division. Recall chapter number 3 in your last semester. We spoke about, right? Chromatin, chromosomes, right? Yes. So when the cell divides, we need to know how is the DNA present, and when the cell is not dividing, how is the DNA present? Can anyone explain me this? What did we speak about? Histones. I don't know about boys, but I think I explained girls about the histone proteins and the chromatin. Can anyone volunteer if you remember what we learned in the first semester?
Horizontally, like a thread, like beads, around it, there's the DNA.
Yes, yes, dear. Very good.
The histone protein,
Mm-hmm.
when they all coil up, they make a chromatin,
Mm-hmm.
and then they turn into a chromosome, which is to
Mm-hmm. Yes, yes, yes. Absolutely right. Please go ahead. So when the cell is not dividing, what do we have? Do we have a chromosome or do we have a chromatin?
chromatin.
Chromat. Yes, thank you. Very good. Chromatin is a long DNA with a protein to fold molecule. Very good. Chromatin. Absolutely. So when do we have a chromosome?
When we have two chromatin,
When we have, yes,
two
dear.
chromatin joined together, like
When the cell has to divide, at that time we'll have the chromosome. Absolutely. Okay, no issues. I will explain it. Okay, I'll explain it in detail. Okay, so we'll talk about the superstructure of the DNA. In the superstructure of DNA, we know that whenever a cell is not dividing, remember that. If you remember the cell cycle last semester we spoke about, right? So when the cell is not dividing, right, that means when the cell is in the interphase, this is all interphase, right? G1s and G2 phase. So when the cell is not dividing, when the cell is an interphase, okay, what happens? So the DNA of the present in the cell, it looks different. It passes through various stages in the cell cycle. Okay, so at that time when the cell is not dividing, the DNA is you know is it's what do you call it? It's wrapped around, okay, certain proteins, and those proteins are called as the histone proteins. The yellow ones which you see here are the histone proteins, right? So if you see here, the blue one is the DNA. This is like 146 base pair DNA, but your book writes as a 147. So let's go with your book as a 147. Okay. So the blue one, which is wrapped around the histone, we call it a histone core, the yellow one. Okay. The yellow one, which you see here, is called as an histone core. So DNA is wrapped around this histone core, but when the cell is not dividing. Okay, so at that time what happens, this histone core is basically, as you see here, it's an octamer. What is an octamer? Octa means 8, right? Octa means 8. So basically we have 4 different proteins. Like if you remember last semester also we spoke about, this is H1. Okay, then we have H2A. Okay, then we have H2B. Okay, and then we have H3 and H4. There are actually 9 proteins, but H1 is, we'll remove H1 from here because H1 is not present in this core. As you can see, the green H1 is present outside the core. Okay, so that means these 4 which you see here, they are basically H2A is this one, This one is H2B, this one is H3, and this one is H4. So they're stacked together, H2A, H2B, H3, H4, and below also you will have H2A, H2B, H3, and H4, which forms the histone core. And H1 stays outside. Okay, it stabilizes the structure. Now, what is happening? Okay, so this structure wherein the DNA is wrapped around the histone core, it results in the formation of a bead-like structure, which we call as a nucleosome. Look at this. So this is a bead-like structure. If you remember, I told you in the last semester also, it looks like, the necklace, the pearl necklace which you have. And each pearl forms one nucleosome. Look at this. That's how the DNA is arranged, okay, when the cell is not dividing. So if I ask you what is each nucleosome made up of, what will you say? What is each
D-
nucleosome made of? It has two parts. Can you tell me what are the two parts?
DNA plus histone protein.
Absolute. It has a core of histone proteins, right? Which are octamer, like 8 in number, and then we have the 146 base pair DNA wrapped around it. But as per your book, it says 147, okay, as per your biochemistry textbook. It's usually 146 actually. Okay. Any questions till now? So, so why do we need to arrange the DNA in this way? Okay. What is the reason of forming these nucleosomes? The reason is because if you remember, I told you last time also, the DNA is very, very long. It's a very long molecule, right? You know, more than 6 feet, if you have to extend the DNA more than 6 feet, more than your height, okay, or you can say 2 meter long, okay. And in order to arrange such a large molecule within the few micron space, absolutely to reduce the space, absolutely, to basically to save the space to accommodate DNA within the nucleus, we have basically God has arranged it in the form of a nucleosomes so that it can be easily accommodated in the compact nucleus. Are we okay till now? Did we all understand what is a nucleosome? You don't need to go more into detail. This is a linker DNA, and what is the role of the H1. That is all. But yes, of course, you need to know one thing over here. Okay, so when we say histone and the DNA, because this concept we're going to use again in the transcription as well as the replication. So I'm going to erase this. So why they are together? There's one more reason. Okay, histones are positively charged. Remember that the histones are positively. I don't want to go into detail. They're positively, or yeah, or maybe because this concept is there in your book, maybe it might be asked. So why they're positively charged? Because histones are the proteins, and they have the two important amino acids. Okay, the arginine and the lysine. Okay, which are basically basic amino acids. Okay, their side chains are basic amino acids. Okay, so they have positively charged side chains. Okay, so as the histones are basic proteins, so they are positively charged, and since your DNA has, so please pay attention, since your DNA has what, phosphate group, which is negatively charged, so your DNA is negatively charged and your histone proteins are positively charged. This is one more reason why your histones and DNA are wrapped around, why your DNA is wrapped around your histone proteins, right? Because obviously there'll be attraction between positive and the negative. So that is why they are wrapped around each other. Are we okay? So the question says that histones, what keeps them together, right? The DNA wrapped around a histone. So the answer is histones are positively charged. Okay, and the DNA is negatively charged because of the phosphate group. Okay. Now this is the 2 nanometer double helical structure of the DNA. Okay, and this, as I told you, that it says 147, but actually it's 146, but you will go with your book. Okay, 147. Okay, So what happens? So this is like, as you can see here, the DNA is wrapped around the histone proteins. It becomes 11 nanometer in size. Okay, so this is like first order packaging of the DNA. Now, can anyone tell me what will be the next one? What will we get after this? After you have many histones together here, after you have many nucleosomes together here, what will we get after that? Multiple nucleosomes, what will they form? They form the chromatin.
Chromatin.
Chromatin,
Chromatin
yes. Remember
string.
last semester? Exactly. Thank you for remembering that, right? We discussed last semester that when you have multiple nucleosomes together, right, they are stacked together, as you can see here, right? So multiple nucleosomes, okay, they're together, they give rise to something called as a chromatin, right? We discussed that last semester, right? So if the question says that what is the fundamental unit of a chromatin, you will say that nucleosome is the fundamental unit of a chromosome. Or you can say that many nucleosomes together, it gives rise to the chromatin. Are we okay till now? Look at this, the size has increased, it's now 11 nanometer. Look at this, we started with 2 nanometer, 11 nanometer. Look at that, this is 11 nanometer now. Okay, now what happens as the cell progresses towards the cell division? Okay, in the cell cycle, as a cell is moving from the interface towards the cell division, what is happening, right? What is happening that this chromatin is going to condense further. Okay, it's going to condense further, right? So what will happen as it will condense further? It gives rise to another structure called as a solenoid. So please pay attention. So what is a solenoid? Solenoid model of the chromosome says that you will have 1, 2, 3, 4. Let me get another color. It's 4, 5, and 6, right? So you will have 6 nucleosomes. Okay, they are together and they form a helical structure, right? It gives rise to the formation of a solenoid. So you can say that in the solenoid, the nucleosomes, right, they fold upon each other and they give rise to the, don't you see it's in helical structure? Isn't it in helical structure? Look at this, helical structure of all the nucleosomes. But remember, in each turn, you will have only 6 nucleosomes. There was a question, I think we had given this in the last midterm, I think. How many nucleosomes you will have in one solenoid, in one turn of the solenoid? Clear, everyone? Okay, great. So 6. Okay. Look,
Doctor, how many do you have in one turn?
you have 6, as you can count them, if you see here, I just said, right? If you see here, 1, 2, 3, 4, 5, and 6. In one turn, you will have 6 nucleosomes per turn, correct? And you have to remember the order as well, okay? What did we start with, and then what will we end with? We started with the simple short segment of DNA helix, right? Then as we went, towards the higher order, right? We went, then it's a nucleosome, then you have the solenoid, right? Then once you have a solenoid, it doesn't stop over here. DNA is further condensing, right? So these chromatin fibers are further condensing, and after that you will have the loops. So remember this. So when this solenoid will further condense, it will give rise to the loops, right? As you can see here, the loops of the DNA, right? Look at this. It was earlier 30 nanometer in the size, and now it is 250 nanometer. It's condensing further. Okay, and after loops it will give rise to the bands. Look at these are the loops and it will give rise to the bands. Okay, these are actually bands, but as per your book it's a loop, but this is a band actually. Okay, with 840 nanometer in size. Okay, it keeps on condensing. By the time cell reaches to the metaphase, it has passed the prophase, by the time it reaches to the metaphase, uh, your chromosome looks like this is a metaphasic chromosome. Okay, wherein it is not only 840, if you add this together, it becomes almost 1400, almost 1400 nanometer in the size. Look at this, how it has increased in the diameter. So this is your metaphasic chromosome, which is a condensed form of your chromatin. So which is visible, which you saw. I mean, you didn't see that, because you didn't have lab, last semester. But we usually show the chromosomes in the lab to students, right? The metaphasic chromosome, the anaphasic, the telophasic chromosomes, right? That is about it. This is the superstructure of the D. If you remember, this was a diagram which we used in the PHSF, right? Last semester. So number one, we have, I'm just quickly going to repeat number one, we have the first order structure wherein we have a short segment of DNA helix, okay, which will wrap around the histone protein forming the nucleosomes. So number two is the nucleosomes. All the nucleosomes will be stacked together. They will result the formation of they will form the chromatin. So you will have all of them will be chromatin, all the nucleosomes together. Okay, then once we have these 6 nucleosomes in one turn, okay, it will result in the formation of a solenoid. Okay, solenoid is further going to be organized into loops. As you can see here, there are loops which are formed. This will be the number 5 loops. Remember the order. Okay, loops will be further organized into bands. These are the bands as you saw here, right? These bands, right? So, and finally, when you have bands, and finally bands will be further condensed and it will give rise to the last order called as the chromosome. This you can see in the metaphase. All clear, everyone? Was it interesting or boring, or you found it difficult? Very important part of this chapter here. Not interesting. I'll show you a video also, don't worry, I'll show you a video also about it, okay? So give me a minute, I'm going to show a video here, so you'll find that interesting. Okay, give me a minute. I'm going to share a video with you all. Tell me if you can hear the sound of the video. Give me a moment. I'm trying to play a video. Okay, what's wrong with it? Give me a minute. Tell me if you can hear the sound, and can you all see the video? Okay, great.
This animation will see the remarkable way our DNA is tightly packed up to fit into the nucleus of every
You can hear
cell.
the sound as well.
The process starts with assembly of a nucleosome, which is formed when 8 separate histone protein subunits attach to the DNA molecule. The combined tight loop of DNA and protein is the nucleosome. Multiple nucleosomes are coiled together, and these then stack on top of each other. The end result is a fiber of packed nucleosomes known as chromatin. This fiber, which at this point is condensed to a thickness of 30 nanometers, is then looped and further packaged using other proteins which are not shown here. This remarkable multiple folding allows 6 feet of DNA to fit into the nucleus of each cell in our body, an object so small that 10,000 nuclei could fit on the tip of a needle. The end result is that the DNA is tightly packed into the familiar structures we can see through a microscope, chromosomes. It is important to realize that chromosomes are not always present. They form only when cells are dividing. At other times, as we can see here at the end of cell division, our DNA becomes less highly organized.
Is it clear now, everyone? Now, was it interesting, Abdulaziz? You're able to relate it now, right? Whatever you understood, and now when you see the animation, it becomes, pretty much, it exactly sits. Yeah, absolutely. It straight away goes into the mind. And where is my presentation? I somehow lose my presentation. Where did it go? I'm trying to share the presentation. Did I close it? No, I didn't. Okay. So is it clear? Okay, wonderful. We can move ahead now. It's not one of the dividing steps. It's basically part of the cell cycle, chromatin, right? It's part of the cell cycle from interphase going towards the cell division. Okay. So that was the superstructure of the chromosome. Moving further. Okay. This we already discussed. Okay, the difference between DNA and the RNA. A quick review, and then we can move to the next part of the chapter. We are already lagging behind. Okay, so a quick recapitulation: DNA, double-stranded helical molecule in which each strand has anti-parallel orientation. Do you agree? Yes or no? It creates a polarity in which the nucleotide sequence is read always from 5' to 3' end. We'll discuss this once again as we'll discuss the replication of the DNA. Okay, so in the DNA, the way, if you remember, I told you we always start from A and to Z, A to Z. We never say Z to A. Similarly, DNA is always read from 5 to 5' end to the 3' end. We never say 3' to 5' end, right? It's always about 5' to 3' end. Okay. What stabilizes the double-stranded molecule of DNA? We spoke about it, the hydrophobic interactions and the hydrogen bonds. Who will answer this question? Which bond is stronger, AT and GC?
GC because it has 3 hydrogen bonds.
Oh, absolutely. Thank you, dear. Absolutely. Very good. Right. And then we also discussed about the in eukaryotic chromosome structure, the superstructure of the chromosome is comprised of many elements like DNA and the histone proteins to form the nucleosome. We just spoke about that, right? DNA plus core histone proteins, and octamer forms the nucleosome, right? So that further forms the chromatin fibers, okay? And further it forms the solenoids, then loops, and finally loops into bands, and finally you get a last structure called as a metaphasic chromosome, right? Yes. And then finally, what is the Chargaff's rule? What did he say? Chargaff said that, yes, number of adenine molecules are equal to the guanine, thymine, and guanine is equal to the cytosine. That is all what we discussed till now. We'll move on. We can move on to the next part of the chapter, which is basically the different types of the RNAs. So this is something which we already know. I'm going to just quickly repeat this. We know that DNA forms the has a capability of forming its own copy. DNA can copy itself, right? And that is called as a replication. So in this chapter, we'll talk about replication, and the next chapter is about transcription and the translation, okay? But at the same time, each strand of the DNA has a capability to form the mRNA, right? Which is called as a transcription. This is a central dogma, right? And once the mRNA is formed, it travels from the nucleus to the cytoplasm, where it goes to the ribosome, and the same mRNA, with the help of tRNA, right, and the amino acids, it is translated into a protein, chain, right? This is the central dogma of our body, right? Now we'll talk about what are the various forms of the RNAs. I'm sure everybody would have heard about uh, tRNA, rRNA, messenger RNA. Tell me how many of you have heard about these three new types of RNAs, the small nuclear RNA and the microRNA? Do you know anything about the nuclear RNA and the microRNA and the interfering RNA? Or you all know about, yeah, of course not. I know that because they were recently discovered. Yes, not very recently, but yes. So most of you are very much familiar about the tRNA, rRNA, and the messenger RNA. So we'll just quickly talk about different types of the RNAs. Okay. So when is messenger RNA produced? When do we produce messenger RNA? Messenger RNA is produced by the one of the strand of the DNA, right? Whenever we need to form new proteins in a particular cell. And we already discussed about it that the entire genome is not transcribed into mRNA. Only a small portion of genome is copied into the messenger RNA. Okay, give me a minute. I'm just trying to put charger in my system. Okay, yeah. So only a small portion of DNA is transcribed into the messenger RNA. Okay, so this is like the basic difference between the replication and the formation and the transcription. In replication, the entire DNA must be replicated, right? While as in transcription, if you have a longer DNA, only one small part of the DNA is transcribed into the mRNA, which later on forms the proteins, right? So mRNA is produced during the process of transcription. And this mRNA carries the genetic information from our DNA, okay, in the nucleus to the cytoplasm where actually the proteins are made. Okay. So then we have the tRNA. In the last semester in chapter number 3, we spoke about the tRNA, right? So this is the clover leaf model of the tRNA, okay, and this is 3D model of the tRNA, okay. And what is the function of tRNA? Anybody? What is the function of tRNA?
Carries amino acids.
Transports amino
Absolutely. It
acids.
carries the amino acid to the ribosomes where the proteins are actually synthesized, right? If you see here, this is the amino acid attachment arm. At this point, the amino acid is attached. Okay, the charging of tRNA, if you remember last semester, we spoke about it. And this is an anti-codon arm, right? This anti-codon arm codes, it binds with, does anyone remember that? This anti-codon arm binds with?
Specific enzyme.
Specific, sorry, your voice is very feeble.
Specific enzyme.
No, the anti-codon arm does not bind to the enzyme.
To the codons on the ribosome.
Codons on, oh, no.
Codon in the mRNA.
Codons on the mRNA, absolutely, right? So if I give you the structure, for example, if I tell you that in the anti-codon arm, okay, in the anti-codon arm, okay, I have, for example, here, let me erase this. In the anti-codon arm, if I have here uracil, adenine, and adenine is on the anti-codon arm. Tell me what will be there on the mRNA. Tell me the codons present on the mRNA.
A.
Okay.
TT.
Absolutely. Is it correct? Everybody? On the mRNA, what will be the codons on the mRNA? Guys?
Isn't it AUU?
Yes. Yeah, very good. ATT. Do we have, okay, wonderful, very good. So after explaining all these things, okay, tell me, what do we have in the mRNA? Which base is present in the mRNA and which base is not present in the mRNA? Do we have thiamine in the mRNA, guys? So do we have thiamine in the mRNA? So what will it be? Yes,
It's AUU.
it will be AUU. So this will form a bond with the, right? We do not have thiamine on the MR. Remember this. And this is like most of the time students do make a mistake here, right? If the sequence is given, right? So remember, we do not have thiamine in the mRNA. We have uracil in the RNA. Okay, whether it's a tRNA or whether it's an mRNA, we have uracil. Remember this forever. Okay, and then we have at least we have one tRNA, right, for 20 amino acids present. Then in a genetic code, we'll talk about that. We can have, we'll talk about that. You will be confused if I talk about genetic code right now. Okay, so that was about the tRNA. Okay, so each tRNA is specific for one amino acid, and this is the codon recognition site. Okay, wherein we have the three bases. I showed you here three bases here, which are called as the anticodon. And these anticodon, that this triplet of bases is totally complementary to what we have in the mRNA, as we saw here. Okay, so on the mRNA you will have codon, on the tRNA you will have anticodon, and this is very, very important, right? This is a very important point of contact when it comes to the translation of proteins in the cytoplasm. So once we'll discuss the translation, I'm not going to repeat at that time this again, so please remember it forever here. Okay, the anticodon and the codon on the mRNA. Okay. Moving on further about the ribosomal RNA. Okay, for the ribosomal RNA, we also discussed this in the previous semester. Okay, for the ribosome, why do we need ribosomal RNA? What is the function of ribosomal RNA? Yes, ladies?
To produce proteins for the ribosome.
Sorry, I don't know why the voice is very feeble. To
Produce proteins for the ribosomes.
carry proteins. No, what is the function of ribosomal? You're right. You're telling me about the function of the ribosomes, which is absolutely right. To create the ribosome along with the proteins. Absolutely. If you remember last semester, we spoke about ribosomes are made up of two things. They are made up of the ribosomal RNA plus ribosomal proteins, right? Together, these two make your ribosomes, because ribosomes are very important when it comes to the synthesis of the proteins, because proteins are synthesized on the ribosomes. 80% of the total RNA in the cell belongs to the ribosomal RNA, right? It's 80% of it is the ribosomal RNA in the cell. Now, please pay attention. If you remember last semester, okay, we spoke, okay, look at this here. First of all, 35% of the ribosomal proteins and 65% of the ribosomal RNA makes together what? It makes your ribosome. Okay. And now please pay attention. If you remember, I told you last semester that ribosomes they basically have two subunits, a larger subunit and a smaller subunit. Do you remember last semester? Right? So it has a larger subunit and it has a smaller subunit. Now here you need to pay a little bit of more attention here. Here they've also mentioned about the prokaryotes. Okay. Rather, we're dealing with the human body. There was no need to mention about prokaryotes, but since it's there in the book and We might ask, okay? So remember the ribosome. So that's why I brought this diagram here so that, it becomes more clear to all of you. So the prokaryotic ribosome is basically the 70S, okay? And we as humans, we have the 80S ribosome, okay, as a whole, okay? But then I told you that ribosome has two parts. The way, if you remember, we spoke about the burger, right? We have, if you're eating a McD burger, you have a top bun part and then you have the bottom bun part. Similarly, you have a bigger subunit and then you have a smaller subunit, right? Let's try to know, yes, 30 and 50 large for which one? It's very important to know 50 and 30 is for the prokaryotic or the eukaryotic. The question says like this: name the subunits of the prokaryotic ribosomes. So 30 and 50 is for the prokaryotes. So smaller subunit in the prokaryotic is 30 and the larger subunit is 50S. Okay. While as for the eukaryotes, for us, the smaller one is the 40S and the larger one is the 60S. Remember these numbers, everyone. Okay. Okay. It doesn't need to make 60. The Sheila, if you remember last semester, I told you that it doesn't need to make 60, right? It's all about 60 and S is 80, 60 and 40 is basically, it's a sedimentation coefficient, right? When you centrifuge these, ribosomes, I don't want to go into details. So when they settle down, okay, so this is basically a number given to them, okay? So this is a sedimentation coefficient. When you add them up, it's not required that it should come as an 80S, right? It's just a coefficient given to them, how they settle down when you centrifuge them, okay? So remember that, right? Even if you add these two together, they are not coming up to 70S, right? So remember, the entire structure is 70S and the 80S, but when you separate them, these are the numbers which we get. That's how they get settled down when we centrifuge them. Based on that, the numbers have been given. Okay. Yes. So remember the numbers, okay, for the prokaryotes as well as the eukaryotes. Now, what else you need to remember here is that, if you remember, I told you that they are made up of ribosomal RNA as well as the ribosomal proteins, okay? Then what you need to remember in this table, if you see here, this is from your textbook, okay? What you need to remember here is, you need to remember here that the smaller subunit consists of one large RNA molecule along with the 20 different proteins. Okay, the large RNA molecule, if you see here, all you need to remember here is, if you see the larger subunit of the prokaryotes, this one, okay, what do we have? We have here one, okay, and two RNA molecules. No need to remember these numbers, 5S and 23S. Remember, for the prokaryotes, you will have two RNAs, right? As you can see, two ribosomal RNAs, okay? And then in the smaller one, you will have just one ribosomal RNA. This one has one smaller ribosomal RNA. Whereas the larger subunit of the eukaryotes, for us, we have one, two, three ribosomal RNA, right? And then for the smaller subunit, we also have one ribosomal RNA. Just remember the numbers. For prokaryotes, two ribosomal RNAs in the larger subunit, right? For the eukaryotes, we have three ribosomal RNA for the eukaryotes. For the smaller subunit, it's the same in both prokaryotes and the eukaryotes. I don't know. That's what I told you. I don't know why they have given about prokaryotes. It was not required actually here. Okay. So then it's trying to show here in this diagram that, so did you all understand till now? It's just trying to tell you the composition of the bigger subunit and the smaller subunit. That is all. Is it clear? Okay, good. So then in this, it's trying to tell you that, okay, if you see here, okay, if you see here, just trying to tell you that it depends upon, if you want to dissociate, you want to separate the subunits. This is a 70S subunit for the prokaryotes. If you want to dissociate into larger and a smaller subunit, all you need to do is you will simply change the concentration of magnesium. Okay, this is basically trying to show you if you're dealing with the ribosomes in the lab, right? So you simply, to dissociate the ribosomes into larger and the smaller one, just simply use this magnesium concentration, which can help them to dissociate. Okay, and when you increase the magnesium concentration, right, it will further combine them together, and you will get, you'll get the 70s ribosome back, right? Just increasing and decreasing magnesium concentration in the laboratory will help us to dissociate or reassociate the smaller and the larger subunits of the ribosome. Are you okay? Any questions about it? Okay. Okay. So all you need to, as I said, that you need to remember what is the overall what is the ribosome size for the prokaryotes and for the eukaryotes, and then what is the largest subunit is the 50s for the prokaryotes, smaller is the 30s, 60s for the eukaryotes, and 40s for the eukaryotes, and then the RNAs, the number of RNAs, which makes up the larger and the smaller subunits. A quick question: where is ribosomal RNA and mRNA located in the cell? Where can we find ribosomal RNA, and where can we find the mRNA?
In the cytoplasm,
Which one is in the cytoplasm? Both of them?
mRNA in cytoplasm.
So, but if you remember, I told you last time also, mRNA is made in the nucleus, right? From the DNA. By using the DNA template, we make the mRNA. So, mRNA is present not only in the cytoplasm, it's also present in the nucleus, right? What about ribosomal RNA?
In cells outside of the nuclei.
What about its manufacturing? Its manufacturing is again done by the DNA. All types of RNAs are made. This is just to confuse you all, right? These kind of questions confuse you, right? Remember, all types of RNAs are made by the DNA. So obviously they'll be present in the DNA as well as in the cytoplasm. This was just to confuse you all. Is it clear to everyone? Irrespective whether it's an mRNA, tRNA, they are all made by the DNA, right? So they should be present in the nucleus as well as in the cytoplasm. Guys, clear? Everyone? Yes or no? Okay, great. Now we'll just quickly discuss the small nuclear RNA and then we can stop over here. What is a small nuclear RNA? Small nuclear RNA is also found in the nucleus, okay? And then what is the function of this small nuclear RNA? Okay, this RNA is also made up of it has an RNA component and it has a protein component, okay? So first of all, remember that the small nuclear RNA is also called as SNPs. Okay, it is also called as the SNPs. Okay, and the way we spoke about ribosomes, they are made up of ribosomal RNA and the ribosomal proteins. Similarly, they also have a protein component in them. Okay, and so then when they are present with the proteins together, they form the small nuclear ribonucleoprotein particles. Okay, SNRNPs they are called as. Okay, is it clear till now? So we call them SNPs. Okay, we call them SNPs. Okay, now very important, what is the function of these SNPs? The function of these SNPs is splicing. Okay, do you know what is splicing? Does anyone know what is splicing? No. Okay. Splicing is something like, let me show you a diagram first. I think I had another diagram also here. Okay, here it is. Yeah. So if you see here, I think I need a blank page. But anyways, splicing is something like, for example, in your DNA or in your RNA. So please pay attention, very important, right? So this is the last thing which you're going to discuss. If this is your DNA or your RNA, you will have certain coding sequences and non-coding sequences present. Say suppose we have these blue squares, okay, and then we have these green squares. Again, we might have these blue squares, okay, and then we might have these green squares, okay. Let's say the blue squares, okay, they are basically called as the exons. E for exon. Everyone, please pay attention. This is very important. E for exons, okay. And then we also have something called as the introns. I for introns. Okay, now what are exons and introns? Remember, exons are called as the expressed sequences. Okay, what do you mean by expressed sequences? That means these exons are the coding sequences. That means they code for some proteins. So they're coding sequences, the exons. Okay, but what about the introns? Introns are like the wasteful thing, right? They do not code for anything. They do not code for anything. The introns, they are silent areas present in the gene, right? Is it clear to everyone? So what are the coding sequences, exons or the introns?
Exons.
Exons. As you can see here, if you see here, the yellow ones are the introns, which are the non-coding sequences, and the orange ones are the exons, which are the coding sequences in the DNA. Same thing, when the mRNA is produced from the DNA, you will see here the blue ones are your exons, which are the coding sequences, and the yellow ones are the non-coding sequences, right? So this is kind of a raw or a pre-mRNA. What is pre-mRNA? Which is not ready yet to be transported to the cytoplasm. Why? Because it still has these non-coding sequences which we need to remove, right? So what is splicing? Splicing, if you remember, I told you splicing is something which removes the what? Introns or exons? By the action of splicing, what should we remove? Exons or introns?
Introns.
Introns.
Absolutely. By the action of splicing reaction, the introns, the yellow ones, are removed from the pre-mRNA, and then only you will get the mature mRNA. As you can see here, it has only and only exons. So now this mRNA is ready to be exported towards the cytoplasm, right? So this is called as a splicing reaction. This is called the processing of the initial mRNA or processing of the pre-mRNA. It's called as the splicing. Okay, is it clear till now?
Yes, doctor.
Okay, so what happens? These SNRPs, they have the capability of splicing. So the splicing action is done by these SNRPs. Okay, when we were studying about the SNRPs, it gave us, we while the scientists were studying about the splicing in the cells, okay, they actually found that in these mRNA, right, there is these splitting reactions are not catalyzed by any protein. If you remember, I told you that SNRPs have both the things. They have an RNA part and they have a protein part. Okay, if you remember the enzymes chapter last semester, which you would have studied previously, most of the enzymes are proteins, except, you might have read about, except the ribozymes. Ribozymes are the only enzymes which are not proteins. They are RNA based enzymes. right? While scientists were studying about the splicing reaction by these nerves, what did they find? They found that these ribozymes are here, the catalysis is not because of the proteins, okay? Here the catalysis is because of the presence of this RNA. But this is like a totally unknown or an unusual thing, okay? An RNA which has a capability of, which has a catalytic activity like the enzymes. So that is why they called it as a ribozymes, RNA with the enzymatic action, right? So they called it the ribozymes. So these ribozymes are very, very important in the splicing reactions which are done after the transcription. Post transcriptionally these are done so that you can have the mature RNA. Did you all understand about the ribozymes and about the splicing reaction and about the small nuclear RNA? Is it clear to everyone? Okay, I think we'll stop over here. We'll not be able to, we didn't even finish the chapter. Oh my God. Ideally, this chapter was supposed to be only for this week, but that's okay. Okay, we'll continue this chapter next week also. Ideally, we were supposed to start the next chapter the next week, but that's okay. We'll finish this chapter on Monday, inshallah, next week. Okay, any questions?